begin, I would like to extend our heartfelt gratitude to the International Peace Foundation for partnering with Mahidol University in organizing this event. Thanks to the IPF, we are glad to have the opportunity to bring together minds from diverse backgrounds to engage in meaningful dialogue and foster greater understanding. It is through such collaborative initiatives that we can truly build bridges of knowledge and cooperation. Today, we are privileged to have with us a luminary in the field of neuroscience, a Nobel Prize laureate whose groundbreaking work has significantly advanced our understanding of the efficacy for the human brain. It is my distinct honor to introduce our esteemed keynote speaker for this event, Professor Edvard Moser, Nobel Laureate for Medicine. Professor Moser's groundbreaking research on the brain internal GPS system has not only deepened our understanding of spatial navigation, but has opened new avenues for exploring the mysteries of cognition. The themes of today's event, the brain GPS, how we know where we are, quote unquote, is particularly relevant in an era where the boundary between physical and digital space continue to blur. As we navigate the complexities of our modern world, understanding the inner working of our brain's navigation system becomes ever more crucial. The Japan-ASEAN Bridge event series serves as a platform for intellectual exchange, fostering collaboration and dialogue between nations. It is a testament to the commitment of Mahidol University and the International Peace Foundation to promote a culture of knowledge sharing and mutual understanding across borders. As we delve into today's discussions, let us seize this opportunity to broaden our perspectives, deepen our insights, and forge lasting connections. May this event inspire new ideas, spark innovative thinking, and contribute to the collective pursuit of knowledge that transcends geographic and cultural boundary. Once
Thank you very much, Professor Panjong Mahathir, the President of Mahidol University. May I now Morawetz, founding Peace Foundation, to deliver introduction.
สวัสดีครับ ASEAN events was related are held in Thailand and Japan. Voice for ethics can meditate and are series which the 2000 application in the ASEAN region. The program is on the transdisciplinary approach towards peace. Where representatives of science, politics, economy, culture, religion, the media, and youth can meet, share viewpoints into each other, and find mutual ways of understanding and cooperation. The aim of bridges is to facilitate and strengthen dialogue and communication between societies in Asia with their multiple cultures and faiths, as well as with people in other parts of the world, to promote understanding and trust. The events aim at building bridges through local universities in Asia to establish long-term relationships with Nobel laureates in all fields, which result in common research programs and other forms of collaboration. By enhancing science, technology, and education as a basis for peace and development, the events may lead to a better cooperation for the advancement of peace, freedom, and security in the region, with the active involvement of the young generation, our key to the future. This is why Bridges is not designed as a one-time event, but as a continuing process of synergies of now over 2 decades, to make the series of events a sustainable success for Thailand and for Asia as a whole. I'm grateful to our honorary chairman, Anand Panyarachon, and to our partners and sponsors who have enabled us to make Bridges a reality. I would like to thank everyone present today to help us facilitate this program. May it help us to bring about a new culture of peace through So we all look forward to his keynote speech and his important contribution to build bridges. A warm welcome, Professor Moser, andขอบคุณมากครับ
Good afternoon. First of all, thanks to Mahidol University for hosting this event, and also thanks to the International Peace Foundation for organizing the Bridges series and for emphasizing the role that science and education can play in creating peace in the world. So I think one should really not underestimate how science, as Uwe mentioned, can act as a platform for dialogue, because in science we try to solve the same challenges, the same problems all over the world. Everyone is contributing with their little piece, and I think there's no better way to actually work together. There's no borders in science, and I would also like to emphasize the role of education in creating peace, because I do think that education, which is based on science, is the way to have people understand each other and to create a better world. So thanks for doing that. Thanks for letting me part of this series. So what I will do, which I can do, is to share with you some of the work that we are doing in the lab in Norway, which is about how we know where we are and how we get from one place to the other. How are we able to find our way? This is a very basic skill that not only humans have, all animals have it, because if you weren't able to find your way, you would be completely lost. You would probably be eaten by some predator. You would not find food. You wouldn't find a mate, and so on. So it's absolutely crucial to survival, and probably a skill that developed very early in evolution, which is of benefit to us because that means we can study wayfinding in very simple species also, as I will come back to. But let me first step back and say a few words about the big challenges in neuroscience. And then, let's see, there. Can we turn down the light a little bit more? Is that possible? Yeah. So I want to begin by introducing one of the real challenges in neuroscience. And that brings me back to about what the situation was like when I started as a student in the 1980s. So at that time, it was still very much two different fields. So you had physiology, medicine on one side, studying neurons in the brain and how they work, what are the basic properties. Then on the other side, you had the psychologists, and I was trained as a psychologist, studying behavior and essentially escaping away from everything inside the skull because there wasn't much known about it and most psychologists were afraid of it. So the two fields didn't really speak very much, and there was a big barrier between the fields. We knew that, of course, behavior was in, and our ability to think, cognition, is very dependent, totally dependent. It's created by neurons in the brain, but we didn't know how. And the size of the problem is illustrated by the fact that just the magnitude, the number of neurons in the brain, almost 100 billion neurons in a human brain, each of them having connections or synapses to about, on average, 10,000 other neurons. And it is in this network of about 10 to the 15th of synapses or connections that the information is stored and the information is processed in the brain. These are very small synapses all over the brain. How can we make sense of it? How can we obtain the information in these connections and related to behavior? If you are to do that, you can't study a dead brain. You must study the brain in action. So no wonder this is difficult. And just to get us started, I'll play this movie of what it might look like in a brain, of course, very simplified. But you see here the individual neurons. They use electrical signals, shown here in white, to send messages. And most of you, at least those of you who study medicine, or know that there are synapses, and that they consist of chemicals, as you see, are transmitted from one neuron to the other as a result of the electrical activity in the neuron. So in this case, the neuron to the left sent an action potential, went to the synapses in the brain, chemicals are released, they go over to the next neuron, and then induce a new electrical signal, and then so it goes on. So this interplay of electrical and chemical messaging is what the brain does. So how can we relate this to behavior? Well, then I think can go a little bit back in history. So I want to go back to around the 1950s. So this is when the first steps were taken to relate activity in neurons of the brain to our own experience. And in this case, experience means what we experience through the senses. So vision in particular, but also hearing, smelling, and so on. And it made sense to begin at the outer end of the brain in a way, where the brain interfaces with the external world. And I put up this picture of David Hubel and Torsten Wiesel, who got the Nobel Prize, I think it was in 1982. Torsten gets 100 years now, so, and he's still very active. So actually, he should be the next candidate if you manage to get him to travel. But it's amazing. He is still an active scientist. I met him just quite recently, and what they discovered here is that in the visual cortex, which is the part of the cortex that that receive signals from their eyes. There are neurons that respond specifically to lines or bars, stripes of certain orientation. And in this case, you see a neuron. Each vertical line is a response or an action potential in the neuron, and you see that this particular neuron responds to. Is there a pointer? Anyway, that responds to the orientation that is approximately vertical. Thank you very much. And the reason why I include this is just to illustrate the fact that cells in the visual cortex responded to certain elements of visual images: stripes, lines, contours, contrasts, and so on. So they were on the track of finding the building blocks of vision. A similar progress was made later in other sensory systems, and we started to get to a point where the brain could be understood at the level of its inputs. So what you see here is a diagram of all of the cortical areas, parts of the cortex that are involved in vision. Each are a box here, and then the lines that showing the connections between them. And my point is only to show that here at the bottom, in this purple area here, V1, that's where they recorded these cells, and that's where the understanding began. And then I'm trained as a psychologist, and I really wanted to understand the more complicated parts of the brain: thinking, perception, imagination, planning, decision-making, memory, and so on. And of course, you won't primarily find that here at the bottom. It will be far up here, wide, far into the brain. So why not start at the top instead? And that's where our research has taken place. So the work that I will present to you today is about the brain's mechanism for space. I could also have talked about time, but there is no time for that, so I will not talk about time. I will focus on space. Space means, how do we know where we are? How does the brain keep track of our own location? And how can we use that mechanism to find our way from one place, how to get from one place to another place? And to a large extent, space, or knowing where you are, is not really a result of sensory experience. It's not the result of what you see, what you smell, or what you hear. It is something that the brain can do that actually is intrinsic to the brain, and that the brain has evolved independently of putting together what comes in from the outside. It's something that exists prior to experience, and if we understand, if we can use space or sense of location to as a gateway to investigating the brain, perhaps we can learn something about its fundamental principles of operation. In many ways, this is rephrasing what Immanuel Kant said already at the end of the 1700s. So what Immanuel Kant, the philosopher, suggested was that our brain comes prepared to meet the world, in the sense that we filter our experience through space, through a sense of time, and also some other properties like causality, which we know much less about. But we can't really think about the world unless we put it into a framework of space, time, and causality. So imagining the world without space is impossible, because space is something that the brain imposes on our experience. It's something that the brain has there from the beginning. That was the idea. And I will show you that there are some reasons for believing that now, based on the recent data that we have collected. So we know more about space than many other brain functions, and the reason is severalfold. But one reason is that space is actually easy to measure, because you can work on this in animals, so it can be tested in rodents. And in a rat or in a mouse, for example, you can test whether the rat or a mouse is able to find its way in a maze from one end to another. So if there is a reward somewhere in a maze, the rat or the mouse is able to navigate and find that reward. If it finds it, it succeeds, and if it doesn't find it, it fails. So it's very easy to measure. And it's also very convenient because, as I said in the beginning, our sense of location evolved very, very early. So it's similar, likely to be similar in very many animals, and probably in all mammals, it's the same system. So it can easily be studied in simpler organisms. And for these reasons, we already know quite a lot, so that we actually have some building blocks to work on. So where in the brain are the places that are important for our sense of location? Well, in one way, the whole brain is involved, but in some other ways, you can say that there are some brain areas that are more important than others. And I want to emphasize two here. So one is the hippocampus in red, and the other one is the less known entorhinal cortex, shown in blue. So both of these are part of what we call the median temporal lobe. So it's the cortex that is on the side of our brain, so kind of behind the ears, and where the cortex folds in under the brain. So it's an older part of cortex, and the hippocampus, for example, is on the very inside, where it curves inside on itself. An early part of the cortex that evolved fairly early, and it's one of those parts of cortex that it has expanded, of course, in primate and human brain, but other parts of cortex have expanded much more. So this was evolved very early, and these areas are known to be important for our sense of location, because we know from human patients who have damages or lesions in these brain regions. So for different reasons, it could be hypoxia, or it could be that in brain surgery, which sometimes had to be done, for example, in epileptic surgeries. Then those patients, first of all, we know that they have challenges, real problems with memory, but they also can't find their way. So these functions are very linked. But it's not easy to find out how neurons produce space or anything in a human brain, because the access is so limited. There are so few patients where you actually can study the activity at the single cell level. And for that reason, most of this work has been done in animals. So, of course, can we learn anything from a mouse brain? Of course, after all, a mouse brain is very small. But the human brain has something like 86 billion, or almost 100 billion neurons. A mouse brain has only 70 million, and is much, much smaller, so you can see the size difference here. Same with a rat brain, a little bit bigger, but not really much. But nonetheless, it turns out that because of evolution, so this just shows an evolutionary tree for mammals, starting with the common ancestor at the bottom left here, and then branching out, you have the rodents here, you have primates up here, and you have other species here. Then, because of this ancestry, we share very much. And we share, for example, the way that the brain and the cortex is wired. So the organization of the cortex has many similarities. So you have the human brain to the right here, you have other primates, and you have rodents here. And you can see that the organization of cortex is quite similar. It's the same kinds of neurons, same types of neurons. They have the same molecules that are expressed in those neurons. They are all it just gets a little bit more complicated in size enormously. But the principles for organizing the brain are very much the same. And that also applies to the language that the brain uses, namely the electrical language, which is the action potentials, and synaptic potentials. So the action potential and its mechanisms were discovered by Hodgkin and Huxley. So they found they balance each other so that you get a very potential and that lasts less than a millisecond, and that is the signal that that neurons in the brain use. These signals are propagated, as you saw in the video in the beginning, goes through the neuron and it causes the release of chemicals or neurotransmitters at the synapses. So this language that neurons use to speak is the same all over. So Hodgkin and Huxley did very much of their work in squids because they have very long axons and they are very big, so it's easy to study these brain cells. The reason why it is also relevant is that this system has not really changed very much in evolution at all. It's the same language all over. Just like DNA is similar for all species, so the neural codes are the same. So having that in mind, then when it comes to space, then it makes total sense to go to simpler species to understand how space and simpler forms of cognition arise in the brain. And that motivated then John O'Keefe, with whom Edvard Moser, I shared the Nobel Prize in 2014. So John O'Keefe in 1971 started to record the electrical activity in the hippocampus, not in the human brain, but in rodent brain. So you see the human brain again here. It was this red structure that you saw. So this is a rat brain or a rodent brain. So he put very, very thin electrodes into the hippocampus. So these electrodes are so thin that they are have a size, a width of approximately, let's say, 50 micrometers. And that is so small that they can squeeze in between the neurons in the brain. And then at the end of each wire, then you have an area that is not insulated. And that there you can pick up the electrical signals from nearby, from the cells in the vicinity. So you can actually record this activity without really damaging anything of the brain cells. So this is same technique that was used now in rodents, that also is used in some human patients where recording is necessary. When you do that, you can pick up the electrical activity of individual neurons in the hippocampus, and that's what O'Keefe did. And I will then show what he found and recorded. So I'm going to show you a movie. And in this movie, you have a rat here, seen from above. So this is from a camera that is in the ceiling of the room. And then the rat is running around in a box, size 1 by 1 meter. So this is just an open box. And the only thing that happens there is that the rat finds a lot of small chocolate pieces that are scattered around. And the chocolate pieces are scattered around simply to motivate the rat to run around in the box and visit every possible place. And while the rat is visiting every possible place, O'Keefe then recorded activity from a single neuron in the hippocampus. And I will show you what that looked like. So in this video that I'm going to show now, the rat is running around. And at the same time, you will listen to the activity of one single cell. So each time you hear a signal, a pop, then this, that's when an action potential takes place in that neuron. And at the same time, you'll also see a red dot appearing on the screen. Okay? You now hear the cell. Each time you hear this pop, then that is when this cell is active. And this happens at the same time when the rat is walking around. And the reason is that you can do that if that electrodes in the brain are connected through a cable to a computer, that you can send the signals from that neuron. And what you see already now is that this neuron is active only when the rat is in the upper left part of the box. Otherwise, it is essentially silent. And this can be color-coded, as you see in this heat map down here. So blue color means low activity, red color means high activity. And you can see that this particular neuron is active only when the rat was in the upper left part of the box. So the hippocampus contains many thousands, hundreds of thousands of these neurons, and each of them have their own preferred location of activity, their own place field, as O'Keefe called them, and he then called the cells place cells, because each of them are active in a certain place. But when each of the cells are active at a different location, that means that if you have access to the combined activity of many cells, you can infer from the combined activity where the rat is at any given time. And that was what motivated O'Keefe and his colleague Lynn Mahidol in 1978 to suggest that the hippocampus was the basis of some sort of map that we have in the brain, because you can read out from the activity where the animal is at any given time. So that was in the 1970s. But there was a big mystery there, because these neurons in the brain that signal location, how can they do that? Because this is not something that the brain infers from the combination of sensory inputs that comes in through the eyes or the ears or the hands or whatever, or the nose. Because you can't just add it up and find your location. It's something that the brain already has from the beginning, probably. So what was the origin of this signal? One clue came about 10 years later, when another group, shown here, is the group of Jim Rank and his student Jeff Taube in the late 1980s. They recorded from a related area, somewhat outside the hippocampus. It's not shown, but it's at the top up here somewhere. They found cells whose firing rate, whose activity was dependent on which direction the animal was turning its head. So some cells were active when the rat was looking that way, to the left. Other cells were active when the rat was looking to the right, yet others active when it was looking forward, and so on. So there was also a directional map with different cells being active in different directions. And this came then on top of the location map in the hippocampus, where different cells were active at different locations. Obviously, these two probably interact for the brain to make sense of where it is at any given time. But not much was known, and that was the basis for our own work in my own lab, where in 2005 we started recording from a third area, the blue area here, the entorhinal cortex. Again, with rats running in boxes, like shown here. And what you now see, I don't have sound on this one, but what you see here is a white dot appearing each time the rat is in a certain, each time the cell is active. So this is a cell from the entorhinal cortex. Each time the cell is active, you see a white dot appearing. And you can see that in this case, the cell is not active only at one place, like in the hippocampus, but it's active at many places. And those places tend to form clusters. So clusters of activity areas. And those clusters, as you can see from the map to the right here, actually form a very regular pattern. So what you have in the background there in gray is the path that the animal has walked over 30 minutes. It visited essentially every possible place in the environment. On top of it, in black, you have all the locations where that particular cell was active. And what you can see is that those locations form a pattern, that it's a triangular pattern that repeats itself all over the environment, or hexagonal if you want. It depends on how you define the unit. But this repeating pattern is like a grid that covers the entire available space. And for that reason, we call those cells grid cells. And the discovery of these cells then formed the basis for the Nobel rewarded discovery. So how does this grid cell work? Well, that was still a mystery. So how could you generate such a regular pattern in the middle of the brain? Because there's no grid in any of our sensory inputs. We don't have grids in our eyes or in the ears or anything. So how can the brain make this? And that has been the basis for much of our work. But one of the first things that we discovered was that these grid cells actually mix with other types of cells, including the head direction cells, the same as you saw, but here it's plotted in a different way. So here you have orientation, 360 degrees, and the curve simply shows the activity of the cell as a function of direction. And this cell, for example, is active only when the rat is pointing its head in the northwest direction, northwest here as well. And other cells then were active in other directions and so on. And these cells, again, existed also together with yet another type of cell that responded to the speed of the animal. So those cells we call speed cells. And you can see some examples of them here. So you have 7 different cells, each shown in color, in a different color. On the x-axis, you have time passing from 0 to 120 seconds in this case. On the y-axis, you have the activity of the cells. And in the background, in gray, you have the speed of the animal. And then in color, you have the activity of each individual cell. And you can see for several of these cells that the activity, like in the yellow one, for example, really very, very strongly follows the speed of the animal, which is shown in gray. So these cells are like speedometers. They measure the speed of the animal at any given time. They are not dependent on location, but you can plot their activity as a function of speed. So here, for example, you see one cell, we have speed on the x-axis, activity on the y-axis, and you can see there's a linear increase. So the faster the animal is running, the more active the cell is. So here you have cells that respond to direction, you have cells that respond to speed, and you have the resulting grid cells. And in that sense, you can make an analogy to a GPS, because what characterizes a GPS is that a GPS as an instrument tracks your own location, the change in location based on change in direction and speed, but doesn't care about what it really looks like at the places where you are at any given time. And so a GPS doesn't bother whether there is a yellow or a red house. That doesn't matter. What it bothers about is how far you have moved in X and Y directions. And in the same way, the grid cells keep track of where you are in X and Y based on inputs that each cell gets that reflect how fast the animal is running and in what direction it is running. But these cells coexist together with other cells, so that includes cells that we call border cells. So these are cells that are active at specific borders or boundaries of the local environment. So in this case, red means high activity, blue means low activity. So this is activity of one cell when a rat is walking in a box that has a rectangular shape. And this cell is active only at the east side of the box. And if you stretch the box the other way, it's still active on the east side. If you insert a wall into the box here, then it's active along that wall too. So it's a very different way of responding that responds to the location or local boundaries. And yet other cells respond to the location of objects, like for example this Lego tower that is put into environment, and the cell then fires on the left side of that object. And if you move the object, then the cell continues to fire on the left side of the object. So these cells respond to locations defined by very significant objects instead of using the grid pattern, which is more determined by changes in speed and direction. So in many ways, you can say there are two systems of two location systems in the brain. So you have on one hand the places which are introduced first because they were discovered first. So it turns out that the map of place cells is very different in every single location. So that means that in this room where you're sitting now, the many thousands of place cells have a relationship that reflects this location. So this corresponds to the role of the hippocampus in storing memories, because it has a unique signature for every possible place that we experience. And this is very different from the map of grid cells and also the other cells in entorhinal cortex, because the grid cell map is just one single map that is applied over and over again in every possible location. So, for example, if you are in this room, two cells that have activity fields or grid fields at the same place, so they are overlapping, having the same clusters of fields. In this room, if you then go outside to the hallway or you go out of the hotel, the same two cells that are active in the same place will continue to be active together when you go out. So every possible place, you only have one single map, and you just calibrate it differently to the environment. Whereas, again, in the hippocampus, you have completely uncorrelated maps that reflect are a signature of every possible environment. So if you have experience from, let's say, 10,000 environments, you're likely to have 10,000 different maps. All right, so then you may ask, is this something that exists only in rats and mice, because all the initial work was done in rats and mice? And the answer is no, because while the work was done initially on rodents, which is one branch of the evolutionary tree for mammals here, later grid cells, place cells, direction cells were all found in bats, and bats are on a completely different branch of the evolutionary tree. And since it's so similar, it means that probably the grid cells or the space system arose very early in evolution. And consistent with that idea, you then also have them in monkeys and in humans. So in humans, we know that because there are humans who have electrodes implanted in the brain, usually because they're preparing for epilepsy-related surgeries where you have to remove brain tissue and you want to monitor the tissue before you decide what to take out. And then for free, you also get then the signals from the neurons in the brain. And it has then been shown that grid-like signals exist also in human brains. So this is the status of what the field was like during the years after the Nobel discoveries. But quite recently, there has been a change in what we actually can do. And that change is that now it's possible to lift our understanding from studies of single cells, one at a time, to the level of thousands of interacting cells. And that's how the brain works. The brain works in networks of cells that all the time influence each other. So you can't really just study them one at a time. Before we had to study them one at a time because that's what the technology allowed. But now we have probes that instead of picking up signals from maybe 3 or 4 or 5 cells at a time that you could still track apart, now you can collect signals from many thousands of neurons at the same time. And the reason is a completely new way of making probes. So this shows one such probe. So what goes into the brain is the thin part that you can barely see here, and the rest is just something you put outside the brain that you connect to the rat skull or the mouse skull. And what you see here is an illustration of what the probe looks like in the brain. So each square here is a recording site where you can pick up the electrical activity of cells that are nearby. So instead of only recording the activity at the end of a wire, which was the old-fashioned way of doing it, now you have actually chips that go into the brain, have thousands of spots where you all can track the activity, and then you get signals from many thousands of neurons at the same time, and you can then study their interaction. And this shows what it is like then. So this shows grid cells again. So each square here is a grid cell. And in yellow, you have high activity. In blue, you have low activity. And again, this is a rat that is running in a square box seen from above. And you can see the grid pattern for all of these cells. But the interesting thing is now we have the opportunity to look at the combined activity of all these cells, not one at a time, but all together. And one way you can do that is to begin by plotting the activity of a subset of these cells. So in this case, it's 149 cells. Each row in this diagram shows the activity of one cell. So black means the cell is active, white means the cell is not active. And then, so this is over a time of a few seconds. And then you can ask, what is the combined activity at any given time point? So take, for example, this slice of activity in a and in case you have 149 grid cells now, then if all these cells were, oh, there it comes. Okay, let's go back. If all these cells were independent, you would have to plot the activity in a 149 dimensional coordinate system. Because they're all independent, they all have their access, and you could plot. Very hard for us to imagine what activity. However, because these neurons are actually quite correlated, what you see in one neuron very much is reflective of what happens in another neuron. That means you can actually break down the activity from 149 to just a few dimensions, and you can still explain most or much of the activity. So these are techniques that are called dimensionality reduction techniques. Principal component analysis was the parent one, and now there are many, many different ways of doing it. But if you now break down the activity only to 3 dimensions, still explain most of the activity, then you can make new plots of the activity. And in these plots, each dot here in this point cloud, each dot is the combined activity at a given time. And over the time of, let's say, 10 or 30 minutes or so, you get a, for each time point, you get a dot, and then you can see the collective activity over this experience forms a structure, and that structure, in this case, is a torus or a donut. So as the animal moves around in the environment, the activity moves around on a shape or manifold, as we often say, that has the shape of a torus. And this is just seen, the torus seen from different angles, and the traces here are the activity as they move around on that torus when the animal is moving around in the environment. And then you can ask, well, is this torus? Because not all the point clouds are so nice as this one, so you can quantify it. There are ways to count the number of holes mathematically in n dimensions, but if you expect to see a torus, there are some rules for what you should expect, namely that you should see one connected component, so one structure. You should see 2 holes in 1 dimension, so that those are shown, the red ones, which are orthogonal to each other. And then you should see 1 hole, which is in 2 dimensions, which is the inside of the torus. It's a hole in the torus. And that's exactly what you find when you look at the activity from an example animal here. So this is just examples of the combined activity of many grid cells when a rat is running around in the environment. The length of these lines show the size of the holes in the different dimensions. So H1 is the number of holes in 1 dimension. So you have 2 holes in 1 dimension and 1 long line or 1 hole in 2 dimensions. So that's exactly reflective of a torus. This happens when the rat is running around in a box. It happens when it runs around in a different maze, even if you don't see the activity of grid cells so clearly longer. And it even happens when the rat is not running at all. It's just sleeping. There are 2 kinds of sleep. It is REM sleep when the rats are dreaming, and it is slow wave sleep, which is the other type of sleep, which when it's very slow brain activity. So those are two main states of sleep, both in rats and in humans. And as you can see, even when the rats are sleeping, you have two long lines in one dimension, one long line in two dimensions, which means that the activity still moves around on this torus as sleeping. But you can still see that relative to the other cells, the activity is drifting around on a map, has exactly the same as in important. It means that the brain has the shape of a torus, and the internal map geography was some 100 years ago because it was the Earth is spherical. So you never get to the end. You just go round and round, and that's the same for the internal map of space. It's also circular, or has the shape of a torus, so that you never get to an end. You just go around on the same map, and you keep track of where you are relative to where you were before. So if the brain map has the shape of a torus, of a donut, then is this something that we learn, or is it something that is there from the beginning? So it's the classical nature-nurture question. Are we born with it, or do we learn it? And it turns out from experiments that we are doing on very young rats that it is pretty much wired up before we have any experience. So this shows first data from a young rat that is 15 days old. At all at that age, but you do see the grid pattern already there. So two long lines in one there. Okay. But the same happens when they're just those brains. We again see one long line in two dimensions, two long lines in one dimension, which means that you have this torus shape. And that happens even if the animals have not opened their eyes yet. They're still closed. They don't open until day 14. And the ear canals are also closed. So really very much isolated from the outside world. But nonetheless, the torus is already there. So that means that the spatial map is there already from the beginning. All right. So now I have spoken 45 minutes, so I'm approaching the end, but I want to make a few points. And that, first of all, then, this is about the development. So I already made the point that this brings us back to Immanuel Kant, namely that the way we perceive the world in spatial terms is actually something that we are born with, and that that although it does not appear until about 10 days, but 10 days is the age when you start to get the relevant connections in this brain area. So before the cells are connected, of course, you don't have any meaningful activity. But when those appear, it's already there before the rats have any experience with finding their way in any way, really. So then one can ask, is there any role for experience at all? so to try to address that, we trained animals, we raised animals in 3 different environments: a sphere, a cubicle box, and a very complicated environment. And it turned out then that those who grew up for 2 months in a sphere and they had they never saw outside. So when we had to change the bedding and change food and so on, were all done in complete darkness. Then those didn't really have very nice grid cells from the beginning, which is different really from those who have to do in the other environments. But they recovered very soon. And here's what you can see. After a few days of training, you start getting the grid cells in those animals too, which means that although if you raise a rat in a spherical environment, it doesn't really have any experience with moving related to the outside world, no boundaries, nothing to relate to. They catch up very, very fast. And that means again that the sense of space is already to a large extent pre-wired, and you need a little bit of exposure to the external world, be able to orient your maps in the right way, but then it's done. So, and this makes complete sense, because if it were the case that an animal or a human being grew the risk being taken by a predator, another article that the animal can. It's probably a signal that I should finish very soon. But I'm getting to the end, and I just wanted to show you at the very end that while I now have talked very much about recording activity with electrical signals, you can actually also watch them through microscopes. And I want to show at the end some work of Wei-Jian Chong, who has been a postdoc in the lab for a long time, who devised a very small 2-photon microscope where you can image the activity of thousands of cells at the same time. And I will show you, skip that. This is the microscope, weighs 2.5 grams. Okay, here. So this shows the activity while a mouse is running around in a box to collect the chocolate pieces. Then you can see the activity of different cells, 4 planes in the brain. Each cell is this white dot, and you see them blinking as the activity goes on and off in the different cells. And you can then follow the activity of the whole brain network. At the same time, as the animal, the mouse is just running around picking up these chocolate pieces. And there's another example here that the mouse is running on a vertical box, so it can climb and it's not disturbed by having this cable here at all. And it picks a piece of biscuit that it finds on the top of the tower. And at the same time, you can monitor the activity in cells in the entorhinal cortex. And even if the mouse is jumping down, which it will down very soon, you can there, and then it goes up again. At the same time, you can still follow the cells without any artifacts of motion. So that means that there are now many technologies where you can monitor the activity of these cells. I'll skip that. And there's then just come towards the conclusion of this work, which is then that the brain does have a GPS-like system for tracking our own position in space. It contains specialized cells like the grid cells, which map our own activity in a coordinate system-like manner. And collectively, hundreds to thousands of cells define a map that has the shape of a torus. It means there's no torus inside the brain, but if you plot the cells along the dimensions that explain most of the activity, you will see that it is a continuous periodic map that never comes to an end, but just goes around and round and round. This seems to be the as soon as the brain develops. And then the final question is, does this have anything to do with diseases where human beings are not able to find their way, and the classical example is Alzheimer's disease, because getting lost is one of the first symptoms, along with poor memory. And it turns out that in Alzheimer's disease, one of the first areas, perhaps the first cortical area to be affected, is the entorhinal cortex, which is shown here in a map that illustrates by color the areas that first start to shrink, where cells die in the human brain, many years actually before symptoms are expressed in behavior, so that you can't find your way, you start forgetting and so on. Then you already get the shrinkage in the size of the entorhinal cortex, which is reflected in the loss of neurons in this brain area. So entorhinal cortex is critical for the early stages of Alzheimer's disease, and I think by knowing that what the cells do there, how they work, it is maybe possible at some time to find the mechanism that start this process that leads to Alzheimer's disease, and perhaps then an intervention that may stop it. I don't need to remind anyone about the magnitude of this problem, but while currently almost 100 million people worldwide suffer from Alzheimer's disease, if there is no cure, then in 2050 it's estimated to increase to 150 million. And the reason is, of course, that Alzheimer's is age dependent. So once you pass about 80 years, the percentage increases enormously and passes 20% of the population at least. And since we know the population gets older, then this is likely to become a major problem, even more on a worldwide basis that requires a solution. So with that, I just want to acknowledge all the people who contributed to the work. So most of the work done together with my Edvard Moser, who I also shared the prize with, and then a lot of people who are listed here who contributed to the various projects, and also funded, of course, by a number of agencies, funding bodies that made this work possible. So with that, thanks a lot for staying attentive, and I again very happy to be part of this series and illustrating one area of science where an international effort has going across borders of all kinds of countries has illustrated how science can be used to create bridges between countries in a peaceful manner. Thank you very much.
All right, thank you very much, Professor Moser. And I believe that Professor Moser is very happy to take questions. We're going to have about 30 minutes for the Q&A session. For those of you who would like to ask questions for the on-location audience, please raise your hand so that one of the staff members will bring you the microphone. And prior to asking your questions, please kindly state your name and maybe your affiliation as well. And for those of you who are online, as for our online audience, please use the chat box to ask the questions. And of course, I would like you to please state your name as well as your affiliation. So I'll start with the lady back there, please.
So good evening, everyone. My name is Prajwal, and I come from Mahidol University International College. So Professor, thank you for the wonderful presentation you've given us. I was really intrigued when you said that the torus structure, can be identified during early postnatal time during the rat. I was interested, was there further study in when the rat no longer showed any movement? The time when the rat probably died. During that period, is there still the torus-like structure in the brain? Is there still activity going on?
Yes, so if I understand your question, over a lifetime, where if there is a time at you mean at the end where there is no activity on the torus any longer? So that activity, as far as we can tell, exists over a lifetime. So it is, of course, we haven't in reality followed very, very old rats. And I think, so rats don't develop Alzheimer's disease, but there are changes with very old aging in rats as well. And what has been reported by other people in places is that the brains of extremely old rats, that means 2.5 years old, lose the ability to distinguish maps that are used to create for different environments. When it comes to the grid cells and themselves, they have only been studied at the individual level, but also there it seems to be some change, but that doesn't necessarily mean that the torus isn't there, because it could just be that the old rats maybe have poor vision and they're not able to really anchor or calibrate their maps to the environment any longer. So it's more like sleep that is just drifting around. When it comes to the early development, it seems like I showed in the talk that at least down to 10 days of age, you will find the torus. And then at some point when you go even earlier, you will not find it any longer, simply because the connections between cells have not been made.
Thank you so much. Just to add on a little bit more, because you mentioned the grid-like activity is still there when we are awake or sleeping, right?
Yep.
Is it still there when the rat is dead?
Oh no, it's not there. When a rat is dead, it's not there, and simply because the cells will stop being active, right? So they don't fire action potentials any longer, at least very soon after the death. So no one has recorded that activity, but I think I can say that very confidently.
Right. Yeah. Thank you so much.
Thank you. Another question, maybe from the professor right here. The next question will be the gentleman back there.
Hello, my name is Dr. Chailet Pichitponchai from Mahidol University, the Faculty of Medicine, Sirat Hospital. I'm very impressed with your findings. I have a couple of questions. In terms of input process output system, it is clearly seen that the grid cells are considered the processing unit. I wonder what are the input or the sensory perceptions that the rat perceive, for example, vision, hearing, equilibrium, the proprioceptions, or probably by the world world magnetic field. Is it possible?
Yes.
And the second question will be, well, I'll ask for one question. Thank you.
Okay. Grid cells, there are two answers to that. First of all, the grid cells, as we see when they are sleeping, they are still maintain an internal map, right? So if you reference the activity of one grid cell to all the other cells, you will see that it keeps this spatial relation, so that the map is still there, even when you almost completely shut off the sensory inputs. I mean, there's still inputs coming when you sleep. You can smell and you can hear, but it's very, very limited. So that the map activity is not really dependent very strongly on sensory inputs. But you need the sensory inputs to relate the map to the physical environment. So that if you come into this room a second time or the third time, the same cells will be active at exactly the same spots each time. And that's because they make links to the walls, to the ceiling, to the doors, everything. So they all get their fixed place. So it means you have a floating map and you anchor it for each environment. And that anchoring has to take place, and that depends on sensory inputs. Then you use vision. You can also use smell and hearing, but vision is very central. But it's not absolutely critical, so you can use the other sensors too. So if you turn off the lights, it's completely dark, they can still do it. It's just that it's a little bit less accurate.
Is it related to the world magnetic field?
Oh, forgot that. Yeah, sure. So magnetic sense in rats is not thought to be very strong, but in other species, like in bats, for example, and in birds, they have a very well-developed magnetic sense, which allows them to find the direction, and they use that for long-distance navigation. So what I have talked about today is short distance. It's just a few meters, right? How you find your way in the room. But birds and bats, and actually also some mammals, but the birds and bats, they use the magnetic sense very actively to find the right direction. So they know where north is.
Thank you very much. I have another question for clinical related to the Alzheimer's disease. Is it possible to have physical activities for the elderly to prevent them from Alzheimer's disease? For example, they do the step dance or whatever about the physical activity related to the place.
Yep. So there are many things that contribute to Alzheimer, or can actually, I wouldn't say prevent, but can at least delay it or make it less likely to happen very soon. One of them is activity, physical activity, yes, but also mental activity, variety, seeking challenges, doing many different things is all very positive, because these things are thought to make the network in the brain cells richer, make more connections to each other. And that means then if or when Alzheimer-like cell deaths start to happen, then you have much more to take from. So it will take much longer before you need the breaking point where you can't operate any longer. So I think seeking an active life, mental stimulation, there's no particular way you can do it in many hundreds, thousands of different ways. Each one has to find their own way. But I think quite the opposite is often depression, right? Is often taken as one out of many, many indicators that can lead to Alzheimer. And that's because you isolate yourself, you step out of social networks, and these things, if it goes on for a long, long, long time, then that could make the brains more vulnerable. It's not the cause of the disease, but just makes it more or less vulnerable.
Thank you very much.
Thank you. The next question, yes, please.
Hello, I'm Zani from Myanmar. I work for an NGO called Community Partners International, and I'm also studying in Mahidol University. My question is a little bit outside of today's topic, but I might not get the chance, so let me ask this question. So my question is that I read your paper on minuscule oscillation. Let me just say the paper is inspired, your latest paper. So my question is that, can the principle of MEC sequence formation be used to develop new learning algorithm for AI systems? And can studying these sequences shed light on emergence of consciousness in AI systems? Because as you know, like AI technologies and a lot of the AI technologies are still based on neuroscience advancement. So I'm very invested in this kind of research as well. Thank you.
Now, thank you so much for reading the latest paper. That's impressive. That paper came out in sometime in January, so I didn't even talk about that. But so that paper was about the formation of sequences in the brain, or how does the brain keep track of order, how A happens before B, which happens before C, and so on. So it turns out that not only is there an internal torus that we use as a base for a spatial map, but there's also some structure that makes formational sequences much easier. And the question then is this knowledge about how the brain works useful for generation of or for the development of AI, or I would just say for developing smarter computers? And that relates to the field of brain-inspired computing, or neuromorphic computing, as it sometimes is called, where the idea is that the brain is a very advanced computer, and we might learn from the brain how the brain actually solves problems in actually to extract some of those principles to make better computers. So there are many things the brain cannot do as well as a computer, and especially speed, because the brain is really slow in many ways, whereas a computer just needs seconds and finds all information it needs. But on the other hand, the brain is extremely efficient, and it works in parallel. Many systems work both in parallel and interactively, and can solve problems that only quite recently were solvable by computers. So like recognizing a face. I mean, we don't need many microseconds to recognize a face normally. And not only human brain, also a mouse or a rat or a cat or a dog can do that. It doesn't take any time at all. So the fact that the brain can do these things so fast by neural networks that it has, and do it with so little energy. I mean, it's often said that the brain operates at approximately 20 watts or something like that, whereas to do similar tasks, a computer does these megawatts. So it's completely out of scale when it comes to energy use. So perhaps we can find out something about how the brain works that makes it so efficient that we perhaps can use for better computers. But I think in general, AI and brain science are likely, like every science, going to interact much more closely, because AI is a tool that will be very influential on the way science is developed. But at the same time, we also need to know what AI does, because there are also pitfalls and there may be ethical dilemmas and all that. So it's like any other tool that you need to control it, you need to know what it is doing, but it will simply make our life much easier, and we can find out much more than without it.
Thank you. Before we go to the next question, there is a question from our online audience, which is quite interesting. Do people whose job is driving, like bus or taxi drivers, have a better place of grid cells?
Yes, at least it was like that before. And the reason why I'm saying that, there were studies done before the time of GPSs, where there was especially a famous study on London taxi drivers. So they used to learn all the streets, part of their training, and then they navigated by remembering all the streets and finding the quickest way. And then in this study, they monitored the activity and even measured the size of the hippocampus, and found that it was bigger and more active in those London taxi drivers than in just a control group. So in that sense, yes. But as we all now know, is that the way we navigate is changing, and the London taxi driver now just needs to press the address and the GPS and go passively. So who knows? Maybe there is no difference any longer. I don't know.
All right, thank you. So we move on to the next questions, please.
Thank you very much for the presentation. Very wonderful. My name is Penol. I'm from Nigeria. I'm a graduate student, Faculty of Pharmacy, Mahidol University. My question is, since sensory input is generally not really a factor for this Taurus like GPS movement, my idea is what is the application, future application, like for someone that is born completely blind, he has no experience of like senses.
Yeah.
So is it the same GPS map? Is it the same way he senses movement? And in the future, what application can we put this? Can people that are born blind be like in signals around them? They can be able to navigate even without seeing.
Yeah, I would say very much the same. That would be my guess. And although we have never studied grid cells in blind rats, for example, or mice, but I think I can say that from the fact that the GPS, or let's say the torus that I showed you, is there already before the animals have opened their eyes, before they have opened their ear canals, so that the system is there. What is lacking early in development is that you need to calibrate it. You need to apply it to the environment. so that it becomes stable. That, of course, vision is very important for doing that, but it's not necessary. So if you don't have vision in a blind animal and a blind human being, they will compensate and do quite well with other senses instead. So, for example, there are studies in humans who are blind from birth that the visual cortex actually can be activated instead very much by tactile finger inputs. It goes there and activates those brain areas instead. So if you don't have vision, you will compensate by developing a richer cortex for the other systems. Of course, it doesn't completely replace it, but the navigation system is so robust that if you lose one input, you just use one of the others, and you can do pretty well still.
All right. Hi. There's a question back there, and then Professor Naratapam. Yes, please.
Thank you for your lecturing. I'm Tanisha Dankijalan. I'm a first-year medical student at Mahidol University, Sirat Hospital. And I'm curious about if you said that the Alzheimer's disease is related to the grid cell because they can remember the map and localization where they are. Does the grid cell correlate with the memory brain cell like the prefrontal cortex that is linked to the ability to learn? Because as we know, the Alzheimer's patients have a problem about their learning skill.
So could you say again the last part? In prefrontal cortex, then we know that what?
I said that does the grid cell correlate with the memory brain cell like the prefrontal cortex? Because Alzheimer's patients always have a problem with their learning skill.
Yeah. No, so key symptom of Alzheimer's disease is memory loss, as we all know. It's less well known that the ability to find one's way is very early affected as well. But they are linked, and the reason is that the entorhinal cortex, which I talked most about today, is very important for our ability to know where we are, and also to track time. I didn't talk about time, but space and time are important. But the entorhinal cortex, those two parts of entorhinal cortex, they connect then at the next stage to the hippocampus. Hippocampus has the play cells, but hippocampus is also very important for memory formation, so that if you have a damage to the hippocampus, then the key syndrome is that you can't remember, you can't form new memories. Very old memories maybe stick, but the new memories cannot be formed. And those two are related because the kind of new memories that you can't make when you have damaged the hippocampus, which is also the case in Alzheimer's disease, is memories that we call episodic memories. So those are memories of things we experience. So for example, how did you get to work today? What did you eat for breakfast this morning? Things like that, the natural things that happen all the time. And those memories always contain a location, they always contain sequences, things that happen in order, so that when space and time and memory, they are very much part of the same system, and when they're damaged together. So when Alzheimer's begins, it usually, you see the entorhinal cortex, in fact in the area of lateral entorhinal cortex, it spreads to the medial entorhinal cortex where you have the grid cells and so on, and at the same time spreads into the hippocampus, so that at the very early stage, already then the memories are affected. And then it spreads further to the rest of the brain and the medial and the prefrontal cortex, all that comes in. But not in all cases, in very many cases, it begins in the entorhinal cortex, and then because the entorhinal cortex and hippocampus are so important for both space and memory, those are affected together, and that's why that happens so early in Alzheimer's disease. Yeah.
Thank you. We have a question from Professor Naratapam, please.
Thank you for the wonderful talk. And I'm curious about adult neurogenesis. Can the process of adult neurogenesis in rodents can replace the loss of grid cells in the elderly in the aging
Yes.
process? Is it possible?
You mean in the normal elderly or?
In maybe Alzheimer's disease,
Yeah, the loss of grid cells.
loss of grid cells, can the process like neurogenesis can replace the new cells?
Oh, if neurogenesis can replace. Oh, yeah,
Yes,
that's a good question. So neurogenesis, as you know, in the adult brain, it doesn't happen in most places, but there are two exceptions. It is in the olfactory bulb, which is used for smelling, and it is in the hippocampus, in a sub-area of the hippocampus that is called the dentate gyrus. In that area, newborn cells are still generated. You get new cells all the time, but it's just in this little area of the hippocampus where you still, as an adult, get new cells, but it's much less than it is in childhood when the brain develops. So that's not enough to replace those cells, and especially because in Alzheimer's disease, the degeneration begins in the entorhinal cortex, so those cells cannot be replaced. So you can still, in this little area of hippocampus, you can supply new cells, but it's not enough to compensate for it.
Thank you very much. I have second questions about other type of vertebrate, like a shark that can use a spatial sense like ampullae of Polysyny to detect the electrical field in marine in salt waters.
Yeah.
So can that signal like electrical field or even the magnetic field from some GI of the bird can integrate into the grid cells?
Yeah, it would be interesting to know. So at the moment, what we know about the grid cells and also place cells is limited to mammals. So we know they exist probably in most animals or maybe all mammals, but whether they exist in a similar form in other species is more uncertain. So it could be, I mean, people are investigating this in birds, investigating it in even in reptiles, but I still think it is to be determined because those brain structures are quite, I mean, there's something in fish and in birds that is similar to what becomes the hippocampus in mammals, but it's also so different, it's wired in such a different way that it's really important to try to find out if there are grid cells, because then they would have to be generated by quite different networks. So I then can't talk about sharks either. And electrical fields, yes, that's another type of sensory input that is special for some species and that we don't have. But I would think that evolution can still use what it has. And if a fish can use electrical fields for localization, then I'm sure that would be reflected in the system. But these are questions that haven't been resolved, and which I think is important to see how different are brain systems before you don't have grid cells any longer.
Thank you. Before we move on to the question from Professor David, as well as the gentleman back there, please allow me to read questions from the online audience. So, greetings from Faculty of Medicine, Sivarat Hospital. Big fan of your work. I was wondering whether these same grid cells and its toroidal manifold could be utilized for more abstract purpose as well. For example, cognitive maps, memory system. If so, could we provide unified theory for both the memory and cognitive deficits, as well as navigation problems seen in these patients?
Yeah. No, that's an excellent question. So what we think about that, and I say think because it's largely speculation, is that the grid cell system evolved early to solve a problem that was pressing, namely finding one's way in space. But the way that evolution works and the brain works is that it tends to recycle what has been successful in some context, and you can use it to solve some other problem. And it is then tempting to ask and think that maybe the grid cell system it evolved to solve spatial navigation problems, but can be used to navigate in other kinds of spaces as well, and abstract spaces, concept spaces. And people have speculated and even shown in human data that perhaps there is a space where you can navigate based on, for example, social networks or the shape of an animal and so on, changes in color, everything. So in a much wider kind of space than just the physical space where you find your location. And the idea is that this perhaps especially happened in human evolution, where we can actually navigate in many kinds of spaces. But the evidence for it is still very little, and it's an attractive idea, but it's still to be determined.
Thank you. Look at the time. We have time for two more questions. One is from the gentleman back there, and the last one will be coming from Johnny.
Okay. Thank you for your talk. My name is Taras from Mahidol University International College. Besides the study of rats' brain activity during their exploration, what about other animals with more complex brain? Do they share the same brain activities during the investigation of the area, or they have kind of different pattern as they explore the area?
So with regard to this part of the brain area that has the grid cells and so on, it is both similar and different. So there has been some comparison between, most of the work has been done in rodents, rats and mice, some work in bats. And then there is work that's been done in primates, in monkeys and in humans. And the monkey work seems to suggest that the spatial map is not really so much based on local motion. So in rats and mice, they use very much proprioceptive inputs, their own local motion, in order to infer where they are, because they get signals from their own movement that tell the brain that now you move so much, now you move so much, and you can add that up and then infer how much you move. But in monkeys, it seems like it's more dependent on visual input and actually where you're looking rather than where you are moving. So this is still unsettled, because in humans, it still seems like the grid cells are able to track where you are. In those cases, they actually move around in a virtual VR environment. So it's not quite the same. So it is still a little bit unsettled. It probably is the case, I would think, that in primates you can do both. You can keep track of your location based on where you're looking, but also where you are moving, because this information is there, and it would be stupid of a brain just to forget about it. So I think everything gets more complex in the primate brain, but the basic elements are there still. And I think this is, of course, just one example, but it's very much the same with cognition in general, that in order to find the basic principles, you can actually see very much of it present already in a mouse brain.
Thank you.
Okay. All right. Well, let's go to Professor David. This is going to be the second to last question. There'll be the last question from right there. Yes, Professor David, please.
So I'm from the Faculty of Science, Mahidol University. So thank you for giving a very general talk. Really appreciate it. And this work is amazing and eye-opening. So it seems that the key geometric property of a torus is to have a periodicity in different lengths.
Yes.
One very short, one very long. And I saw that in your graphs, you're able to track as a function of time where you are on this torus.
Yes.
So do you see any association with different types of movement, like in a along the longer length? Or?
Yeah, that's what I would think, based on what we have seen in studies of place cells before, that to some extent they scale to the physical environment, so that the movement is not absolutely stereotyped. It may be reset or scaled to the actual environment. So that means that you can't really use the speed of movement on the torus as a reference for how far you have moved. You must also calibrate it to the actual environment where you are moving. That would be my guess based on what we have seen in individual grids. But the real experiment to test that idea is still to be done.
Thank you. And the last question, please.
Well, thank you very much, Professor, for your exciting and very wonderful presentation. As a nutritionist from the Institute of Nutrition, Mahidol University, I'm more interested in, this is very good, but I'm interested in a nutrient. Is there have you ever come across any nutrient that can delay the onset of the AD? For example, people talk a lot about cacao. What do you think about it?
Yeah, I don't want to comment on that because first of all, I'm not an expert, and second, there's a lot of hype. So what is popular one day is not popular the next day. But I think in general, Alzheimer's disease is like many brain diseases. It's very much a matter of it has, I would say, it has a component of lifestyle, which includes both how healthy your eating is, how much exercise you do, how much brain exercise you do. But of course, we can't escape the fact that there's also strong genetic factors in some people. And there are certain genes, like, for example, the ApoE4 genes, that in certain gene variants then increases the risk of getting Alzheimer's quite a lot. I mean, about tenfold if you have the really wrong combination. But it is nature, it is genes, and it's environment. And I think as long as we don't really know what causes Alzheimer's, the best thing is just to try to live a healthy lifestyle. But I wouldn't say that there's one single nutrient. I think the evidence for that is still to come. And maybe I'm not an expert, so perhaps you can tell me and I may be wrong.
Thank you so much, Professor Moser. So, ladies and gentlemen, please put your hand together for Professor Moser.